All right, just continuing from our last lecture on glycolysis, we're moving on to pyruvate oxidation. And we just did a little review of, well, what is pyruvate? Pyruvate is a 3-carbon molecule that's basically half of a broken down glucose. Now, it's been modified a little bit, but you don't have to know anything about its chemical structure. So that's really good for you guys. So if we're talking about pyruvate, if oxygen is present, now this is a big if here, because the AP people really like to ask questions about oxygen and respiration. So pay attention to this a little bit, because if the oxygen is not there, some special stuff happens. Basically, the cells suffocate, but there are some things that can block the oxygen. So no oxygen means no mitochondrial function. So if you're suffocating, you can use glycolysis and fermentation for a little while, but that's usually not enough to keep up with the needs of, your brain and your important biological, metabolic functions. So just know that pyruvate can't enter without oxygen. So oxygen has to be present. So O2 is very important here. Okay, so what we have happening next is pyruvate is oxidized. Well, what is it oxidized into? It's oxidized into this intermediate product called acetyl-CoA, okay? And that's used to make a product we need for the citric acid cycle. So for this, that's going to be citrate. So citrate is what you need for the citric acid cycle. It's kind of nice that the name is, just sounds just like the process. Okay, what else do we have going on here? In this step, we have CO2 and NADH being produced. And if one of those is a waste product, the other one we're going to use a little bit more later. So CO2 is the waste product. You know this because everybody knows that you exhale CO2 in regular sort of in respiration at the organism level. So, all right, so pyruvate oxidation, this diagram right here, if you're going to put something in your notes, this is it. We take the pyruvate, just know where it came from. That came from glycolysis. Okay, so that, are my notations showing up and readable?
Yeah, you're good.
Okay, thank you. So we use an enzyme here called CoA. We take NAD and we convert it to NADH and CO2, which is waste, which you can kind of see here. And we get this substance called acetyl-CoA. So this is it for pyruvate oxidation. It's a really simple step. The problem with all this is there, as far as being a student and being someone who has to learn it, is there's so many steps. You're simultaneously learning photosynthesis and respiration. So there's a lot of room for mistakes to be made because there's multiple, you break down photosynthesis and respiration, they have very similar inputs and outputs, but the intermediate steps can be a place that you can be confused, which is why I'm going to give you an assignment to review the basics of respiration today, okay? I know it seems like teachers just give you stuff for the heck of it, but this is some tough stuff, and we want you to practice it a little bit. Okay, so pay attention because we just moved into the next step. Pyruvate oxidation, there it is right there. We turn pyruvate into acetyl-CoA, and we also get NADH. So know what's made in pyruvate oxidation. Know your inputs, okay, your inputs, and know your outputs, which I'm going to underline in green. So know your inputs and outputs. So in and out. All right. That's the big idea. I just kind of want to mentally show that we are shifting to another phase of respiration. I hear somebody's mic off. Does anybody have a question?
Yeah, so CoA is just the enzyme that helps catalyze that.
CoA is what they call it. All right. So we're now in the citric acid cycle. Let's take a look at it. Okay. Also called the Krebs cycle. Okay. So Krebs, citric acid cycle are the same thing. This happens in the matrix. And if you remember, the matrix is the stuff inside of the inner membrane. Okay. So the matrix is this purple area up here that I'm shading in. So you want to know where things are happening. Okay, what is its input and output? Okay, it turns acetyl-CoA, made in this step, pyruvate oxidation, into citrate. Okay, so citrate is an output. And this is where we have a lot of, we have CO2 being released. So know your inputs and outputs, okay? Input is acetyl-CoA, output are citrate and CO2 so far. We also get ATP in this stage, so there's another output of the citric acid cycle. And the electrons that we made in the previous steps are transferred to a molecule we've already been talking about, but we're going to add a new one here. We have NADH and then we have FADH. So we have more electron transfer, So these are going to be considered outputs. So I think I have all my outputs in blue and my
Mr. Sater,
yeah.
Can you slow down a little bit?
Yeah. That's fine. Let me review it because and I need that. When I'm teaching online, it's so hard for me to watch my pace. So again, citric acid cycle, Krebs cycle. We talked about where it happened up here in the matrix, which is purple. The input, acetyl-CoA, which came from pyruvate, or pyruvate oxidation, is turned into citrate. We have CO2 waste. We get some ATP, and we also get out NADH and FADH. Let's take a look at another view of this. Okay. This is the full citric acid cycle. You do not have to know this. I just want you to see how the sausage is made. That means like see all the, ugly details. You're only worried about the input, which is pyruvate up here, and the output, which is citrate right here. If you want to be a biochemist, this is the kind of stuff you're going to know, like the back of your hand. So we don't get into this depth, but I think it's important to kind of peel back the curtain and show you what's really happening. A lot of times in science education, we just say, well, this goes to this without really showing that it's more complex than you might realize. There's a big debate in the AP community that I follow online all the time that says, do I have to teach my kids every step of the citric acid cycle? And everyone always comes back and says, no. But there, I don't know why, but there are some instructors out there that really want their kids to know this whole thing. I don't know. That's pretty sadistic. All right, let's go on to the next simplified version. I like this one a lot better. Okay? In the citric acid cycle, we put in, let me do blue again for inputs, and we get out these things over here. Okay? So if you're going to know something, it's this. Okay? Just add in, where's this happening? This is all happening in the matrix. Okay? This is like, you know, put a star on this in your notes. This is what you really have to know. Now, I am making a video of this. Hopefully the video turns out better than the last one where I was glitching a lot, but I don't know what's up with that. I'm trying to get better at this. Turned off all my windows I'm not using today. Maybe that'll help. All right. I'm going to go on to the next section. But before we do, let's ask you a few questions. I'm sure I probably have the answer showing up automatically. All right. Obviously, I've said it 2 or 3 times. Where does the citric acid cycle happen? It's in the matrix. I want you to know that. Next. What's the net production of ATP? It's 2. If you go back to glycolysis, how many have we made so far?
2.
Well, add these ones on.
Oh.
4.
4.
Yeah.
4.
Okay. So let's go into the final step here, oxidative phosphorylation. Now, this one will kind of look like some of the things from photosynthesis. All right. There's a couple things going on here. We've got an electron transport chain, which we're going to abbreviate ETC. Okay? So the electrons are going to go through multiple steps. And then we have chemiosmosis. We're going to basically set up a gradient. And whenever we have a gradient, things are going to want to equalize. And that gradient is going to spin a pump. And we're going to make ATP with that. Just like we did in photosynthesis. Okay. So what do we get here? If you look at the electron transport chain, it is multiple steps in these transport proteins on the mitochondrial matrix membrane. So if you look at this bilayer here, that is going to be the membrane I'm about to draw in purple. And this is why we have all the surface area on these cristae to get more room for these proteins that make up the electron transport chain. Okay, so what are they doing? They are using NADH and FADH to pump hydrogen into the outside of the cristae. So this area right here in green, we want to put all the hydrogens in this area right here. And then they're going to go through this ATP synthase, and it literally spins it around. And when that spins around from the hydrogens going through it, it makes lots of ATP. So this is where we get an incredible burst of ATP. Anywhere from, depending on what source you look at, 18 to 36 ATP. I don't really care that you know the number. I just care that you understand, like, this is where most of it is made. So what's it basically doing down at that level? It's putting ADP, which you can see right here, back together to ATP. So there's a lot of AP biology instructors will try to get students to memorize all these things in the electron transport chain. It's not required. All you need to know is NADH, FADH power the electron transport chain to get all these hydrogens on the outside. Well, now we have a membrane potential. We have more things on one side than we do on the other. Well, that's going to allow for these hydrogens, almost like a waterfall. They're going to fall back to the other side of the membrane. Which way are they going? They're going into the matrix. So when hydrogens go back into the matrix, they spin ATP synthase, and this is where we get a big boom of ATP. All right, I'm trying to slow down on purpose. Any questions right now?
So it's just using NADH and FADH to power the pumps to be able to put hydrogen on through that goes to that other part, correct?
Yeah, let me say that just so we're going to use NADH and FADH to pump hydrogen out. Then it's going to fall back in, and when it falls back in, it spins this ATP synthase up, which is your big burst of ATP.
So is it pumping out of it to go back in?
Well,
or is it pumping in out?
If you look at this arrow right here, they're coming back in.
Okay.
Okay. Now, what happens to NADH and FADH after they do this electron transfer? The H's are gone. Where did the H's go? They went out here. So these NADH and FADH are basically hydrogen carriers.
Okay.
All right. Everybody okay so far?
So, like, do we need to memorize this whole thing, the whole chain, or is it just like...
No. I'm going to show you what you need to know on the next slide.
Okay, because it's a lot of terms, like...
It is.
I cannot, put it...
Dahlia, I don't know if you remember me saying, but most teachers and most students say this is the hardest unit of the class.
Yeah, it's like all the terms overlap, and it's just different parts of the chain.
They do. You got it. You're basically following things through a factory. Okay, so there's different parts of the factory that do different jobs. The big thing that I want my students to know, and I want them to know this because this is what AP tells me exactly what to teach. I don't need to and in COVID, I'm trying to boil down only the essentials. Because we don't have time to. I mean, it's cool to be exploratory and talk about weird things, but we really have to just get the most essential knowledge down. So let's look at that electron transport chain a little bit. These are things I talked about on the last slide, but let's break it down a little bit. So remember, this is taking it, we're basically reviewing what we talked about already. We have the inner membrane, and that's where the electron transport chain is at. This is that blue line right there. Okay? So, what is it basically? It's a collection of protons, and the electrons fall. And what do I mean by that? They just keep jumping down, and they keep getting reduced and oxidized, reduced and oxidized. And as they do this, that hydrogen concentration builds up. Now remember, you're seeing hydrogen build up, so that's NADH and FADH basically losing their hydrogens. Okay, so this is one side of the final step, this phosphorylation. All right. Now, I already talked about this, but just so it's part of the lecture. Sometimes I get ahead of myself. All those folds on the cristae give more places for these reactions to occur. Cells want to be efficient. You know, usually most people, more is better. Well, this is one of those cases. The more of this I can make more efficiently, the better. Now, the electron transport chain, just make sure you know that that's not what's making the ATP. What's making the ATP is the ATP synthase. Now, this is something that I didn't talk about. The final electron acceptor is oxygen. And the oxygen, all this hydrogen goes somewhere. It bonds with the oxygen, so you get 2 hydrogens, 2 electrons. Well, that and an oxygen makes water. So water is an output of respiration. And if you go back to the most basic equation, it does show water on the right-hand side of the photosynthesis equation. Sugar plus oxygen. Let me write it. That gives you CO2 and water. Well, where's that water come from? It's coming from this step right here. So all these hydrogens, you can see them, right? This is where you see it down here. All these hydrogens bond with the oxygen that you're inhaling to make water. Okay, so already talked about this. The most important function of the electron transport chain is to make that proton gradient. It's to concentrate protons on the inside or outside of that matrix. Where's it concentrating them?
Inside.
Let's go back and look. It's pushing them this way, so outside. So we're concentrating them outside, and then they diffuse through this ATP synthase to make ATP. So by pushing all these hydrogens to the outside, we have this potential. So we have this chemical potential energy. And when they pass through there, that energy is used to make ATP. Now, this is where we're going to give this a name. That gradient, if you have a lot of hydrogen on the outside and just a little bit of hydrogen on the inside, this is going to want to pass through here. Okay, this is why we learn transport first. So all these hydrogen ions are used to do cellular work. Well, what's that cellular work going to make? It's going to make ATP. So the electron transport chain, just to kind of show it to you again, we've broken this down several times. All these hydrogens get pumped out. Okay, what is this? Look, we're inside the matrix down here. You can see matrix. And so we pump them out, and then they pass back through ATP synthase. That passing through is where the energy comes from. I'll try to show you a video. I didn't get it ready. I might have to stop recording and show that to you of like a model of ATP synthase, because it actually looks really cool. Okay. So this section right here, this ATP synthase, that's where we get into chemiosmosis. Using osmosis is things passing through a membrane. We're using chemicals that are passing through a membrane as potential energy to make all this big burst of ATP. So that enzyme is called ATP synthase, and it uses the energy of the hydrogen crossing that gradient to make that ADP into ATP. Everybody okay so far? I'm trying to consciously slow down. Am I doing okay?
Just 1 sec, sorry.
Oh yeah, wait a sec. Sure.
You don't have guided notes for this, right?
I have a packet I can put out to you. I didn't make a formal copy of them because most people want them on paper, but I can put them out and you can use them as a cami if you want. So if you want to take those guided notes, rewatch this and fill them in, that might be a great way to review. I don't want to assign that to you, but if you want to do it, I will definitely make it available.
Thank you.
Hey, Mr. Sater, are we turning in notes again today or no?
No, I do want you to kind of take some notes, but I'm not going to ask you to turn them in as a graded assignment.
Okay.
All right, let me go ahead and move forward. So I want to show you this in a video after I end this lecture. ATP synthase acts like a rotor. Well, what's a rotor? A rotor is like a spinning shaft, almost like a water wheel, if you've ever seen like the old-fashioned water wheels from, long ago. So when that rotor spins, it basically activates sites, so catalytic sites, so it's acting like a catalyst, okay? It's not catabolic, it's catalyst, and it turns your spent ADP back into ATP in very, very large amounts. And the section we're talking about is right down here. Where does this happen? It happens inside the matrix. ADP is in the matrix. Okay, now this is sort of that big idea. It makes about, I think I told you 36 before. I meant 26. I'm sorry about that. It makes somewhere in the range. And again, I don't really care that you know the exact number because there's, variation here. But it makes a lot of ATP. Remember, we made 2 in glycolysis. We made 2 in pyruvate oxidation, and now we're making many times more, almost 5 times as much. So this is where organisms that are needing lots of energy, like big organisms take a lot more energy than like bacteria. So to maintain that sort of metabolism, we need a more complex way to make ATP, and here's where it is. It's cellular respiration. All right, couple review questions for you. How's this proton gradient made? Okay, how's this proton gradient made? Well, what's the proton gradient? Those are all those H pluses. Okay, so what we see happening here is we have an exergonic flow. That means energy is being released. How's that working? NADH and FADH supply the energy to pump hydrogen outside of the membrane into the inner membrane space. Well, where's that inner membrane space? That intermembrane space is this purple area. That's where the hydrogen's going to build up. Then it's going to fall back in. Okay. What's the final electron acceptor? It's oxygen, which leads us to make what? What's oxygen and hydrogen get you?
Water.
That's right. Okay. Just a couple review questions here. All right, last one. Let me move my words out of the way here. What does ATP synthase, how does it get the energy? It's from the proton gradient. It's from what another name for proton gradient is the H pluses. So when here's the membrane, when the H pluses fall back through here, that supplies that energy we need to make that big blast of 20 something ATPs. Okay. This is probably something that you're going to need to have committed to memory right here. So I know I don't want to use names because I'm going to put this on YouTube, but there were people saying like, well, this is so much, and the terms are similar. This chart that summarizes everything, I think, is what you need to focus on. If I could add one thing that I should have added, it would be location. So if you could put down another column that says where things are happening, I think that would be smart. I'm going to leave that up for a minute so you can capture that. You know, you can do screenshots on the meet. I don't care. So make sure that you capture this. Anybody want me to wait? I'll wait, but I'm going to leave this up, but I will stop recording for you people who are watching this recorded.